Good morning. I think we've seen all the parts of hosting a webinar this week. We've been talking on the webinar without noticing it. I made a huge mistake when I had to present my paper and I couldn't get the thing right so that my paper can be displayed. But in any way, bear with us. And today is our last day. So good welcome to everybody. And thank you for all those who participated from Monday till today. The attendance was very good. We had an average of between 47 and 50 participants per day, which is very good. I can just say that the students that participated was about 17, and the number also increased, and the rest of the participants is industry technologists or professionals or consultants who also joined us. Today we've got a very interesting program. We will start with Dr. Neil Fourie, who is a veterinary toxicologist. I won't be able to tell you exactly what that is. I just know that Dr. Fourie has a very impressive career so far, but I will hand over to him so that he can tell us exactly his journey up until now. After his presentation, we've got two very interesting presentations on bovine leukosis, EBL, and then brucellose. And after that, we're going to more the psychological side of being a vet, where you can do the personality trait assessments, and then we conclude with a briefing by Dr. Todd Collins. I had a wonderful conversation with him yesterday, and I really want to inspire you to please stay till the last presentation. But now, Dr. Neil Fourie, I'm going to hand over to you, and please explain to us your career and being a vet as a toxicologist. Thank you.
Thank you, Lena. Yes, good morning, everybody. This is also new to me. I'm used to lecturing at the faculty, especially to the feedlot school. But I drove all the way from Groblersdal to come and sit here at the MPO's offices because our signals are not very good there. Just about my background, I qualified in 1982 with the senior manager here at the MPO, Dr. Chris van Dijk. We were classmates. And then I spent 2 years in the Defence Force, and thereafter 12 years at the OVI, the Onderspoort Research Institute, working in the Department of Toxicology. And that was the best part of my life. You know, we were exposed to, we are, and they still are, and we were the reference center for toxicology in South Africa. So every possible case came to us. And a lot of what you will see today emanated from that period, the 12-year period that I worked for the Research Institute. It was the golden period of the Research Institute. Many things were researched at that period, published, and many new findings were made. After 12 years, I finished my PhD there on Gaussiekte, which we won't discuss today. I isolated the toxic principle. I've got a liking in chemistry, and I spent like 8 years in a lab isolating the toxin from Gaussiekte inducing plants. And then after I left, I joined industry, the pharmaceutical industry, for 10 years, where I developed many products for Intervet, which is now MSD, again, my chemical background. Then I got fed up with industry, and I went to private practice in Middelburg and Mpumalanga, and I've been there the last 10, 12 years in Mpumalanga on the Highveld. Very interesting area. We're on the Highveld, so we see all the diseases, problems of the Highveld. And 30 kilometers away, we're in the Lowveld at Groblersdal in Loskop Dam, where I live. And there we see heartwater and gifblaar and all sorts of nice things of the Lowveld. So it's the most beautiful place to practice is Middelburg. You see literally everything that you can study at OP is available in Middelburg. But yeah, I still practice as a toxicologist. I'm involved in many litigation cases. So we'll discuss that today as well if we have time. By the way, Lena, you must please check my time and we'll have a break somewhere in the middle of this discussion. Yeah, I do quite a number of court cases where they need an expert witness, expert opinion, and it's quite interesting. So the take-home message there is don't go to court. If you do go to court, know what you're doing. Otherwise, stay out of court. And because it's not an easy place to be. Okay, when we think about dairies, obviously we've got animals in milk, and there are also heifers we are raising to replacement heifers to replace these cows. They are natural grazing and that sort of thing. So similar to a feedlot, I always say that the feedlot guys must know everything because that's the point of the pyramid where it ends up. And it's the same with the dairy school. You must know everything from rearing calves to what's going on natural grazing, what's going on in the dairy itself. So you must really be well equipped with knowledge on many, many aspects. Today we are talking about toxicology, the study of poisons. So let's get away from nutrition, from your infectious stuff that you dealt with, and look at the non-infectious causes of problems in dairy animals. So what we're going to start off with is what are animals exposed to? Now, in a dairy, this roughage includes grazing, obviously, as well. Animals graze. So you must know what's going on in the grazing in your area. Talking about area, you must know your area. That's extremely important and what's going on there. So roughage includes grazing. Then obviously animals are exposed to loss of concentrates to improve milk production. And we'll discuss the problems that emanate from that. We use all sorts of feed additives, and we'll see what problems we can encounter there. Water we will discuss very briefly, and then animals get medication. So just keep this framework in mind when you examine a case of problems. I hope there's a program on television, The Mythbusters. So I hope today you will learn a few things that has become like common knowledge in veterinary science and which is not so. And I hope to, especially when we come to fungal poisonings and that sort of thing, we will discuss that. I have got an agreement with Dr. Van Dijk. If you can't write down 10 things in this presentation that you haven't heard before, you'll get your money back. Okay, right. We, as toxicologists, classify 8 plant poisonings as the most important poisonings in South Africa. These 8 syndromes account for about 90% of stock losses due to plant poisonings. Obviously, we're not going to discuss this whole list because there are very few people that milk in the gifblaar area or the gousiekte, so we'll delete that. Or geeldikop, vermeersiekte. I mean, those things occur in the Karoo, but just for interest sake. Those are our 8 most important syndromes. What is of importance to the milk industry is the cardiac glycoside group, and I would like to discuss that very briefly. We've gone through a severe season of toll poisoning on the Highveld and areas of KwaZulu-Natal. So unfortunately, if you deal with toxicology, I must teach you a bit of chemistry. That's very important, and it's my pet subject. You can remember we get cardenolites and bufadienolites. The cardenolites are the Nerium oleanders and milkweeds and all those seriously poisonous stuff that are of no economic importance because they are too toxic and animals don't eat them. They've got on C17 of the steroid structure, they've got a 5-membered ring there. Your bufadienolites on C17, you've got a 6-membered ring. These bufadienolites are the plants of seriously economic importance in South Africa. Where does the name bufadienolites come from? Those of you that study vet science, I'm going to take you back to your third year now. And the bufadienolite name comes from the toad. It's not a frog, it's a toad. And that's a buffa species. And in this parotid gland here that are squeezed, there you can see the poison coming out. And that contains a cardiac glycoside, which is a bufadienolite. So that's where the name bufadienolites come from. You'll see if a dog mouths or licks one of these toads, they will start salivating. And if they take in the skin, they can even die. So toxic these toads are. But this is most probably the most important toxic plant in South Africa, accounting to heavy stock losses every year. Moria pallida, the yellow tulip. There's a close-up of Moria pallida. And look at this. This is in my area on the Highveld where there are several dairies. It loves disturbed land. You can see this is an old land, an old cultivated land. It looks like it's been planted here. All the green that you can see there is tulip. And this is obviously the time of tulip. You can see, look at the background here. It's towards it's the end of winter. And tulip is a winter problem. July, August, September, where there's no green stuff around, we get tulip poisoning. Now, what's the clinical signs that we see with tulip poisoning? It affects 4 systems in the body. Obviously, the heart, because it's a heart poison, causes tachycardia and cardiac arrhythmias. So if you put your stethoscope on that heart, you will hear the severe arrhythmias of these animals. But most probably the most important clinical sign, which sounds insignificant, is this weakness in the hindquarters we can see in this animal. They go semi-paralyzed in the hindquarters, and eventually they go down completely, and they can't stand up anymore. You get a slight bloat because of rumen-atomy. It's a slight bloat. It's not like a bloat that you'll get with lucerne or one of those C. And then you get respiratory distress. You can hear these animals from a distance. They grunt. They go, difficult breathing. Okay, that's tulip poisoning. What are the key words for tulip that you must remember? And that's what we encountered this season. Newly introduced animals. Remember that. Animals that were born and grow up on tulip veld usually avoid tulip. It's newly introduced animals. They will eat the tulip and they will die because it's highly toxic. We talk about a toxic dose of around 1 gram per kilogram body weight. So if an animal like this, which is about 250 kilograms, taking 250 grams, it's dead. Okay, newly introduced animals. The thing you must also remember is that it's a hay contaminant. So if you feed hay, teff bales or that sort of thing, you can get cardiac glycoside poisoning. You must open those bales and look for the tulip leaves. It's a serious hay contaminant. So newly introduced animals and a hay contaminant. It takes about 4 days for animals to get used to new grazing. We had many cases in Middelburg, even in the feedlot industry. They bring in animals from all over South Africa to the feedlots, and they get backgrounded. Obviously, before they go into the feedlot, it's animals that don't know tulip. They go into these natural grazings and they eat tulip, and we had severe stock losses this year. So that's tulip poisoning, a cardiac glycoside poisoning of massive importance. You can see the distribution here. The yellow is the distribution of tulip. So this is in KwaZulu-Natal, right down in the Eastern Cape where our dairies are, KwaZulu-Natal, throughout the Free State, Mpumalanga, and all these areas. These are all the other cardiac glycosides containing plants that we will not go into today because these are all the Western Cape, the pluckies, the succulents that also contain cardiac glycosides. But let's focus on tulip, which is in this whole area and that causes severe stock losses. Now, I would like to test your memory. What's the treatment for tulip poisoning? I'll give you 10 seconds. There is only one treatment, and that's medicated activated charcoal. And the dose is very important. 2 grams of activated charcoal per kilogram body weight. So an animal like that, say it is 250 to 300 kilos, it needs 500 grams of activated charcoal. And what you can see, what we did here, you dose them right there in the veld or on the maize land where the tulp is and where the animal is down. You don't chase it to a crush pen, it will die. There's lots of research that's been done on all sorts of fancy things like lignocaine to stabilize the heart and that sort of thing. Nothing works except activated charcoal. You get reabsorption from this systemic circulation of the toxin into the rumen. And the activated charcoal adsorbs it. It's not absorption, it's adsorption. So activated charcoal is a very interesting compound. It adsorbs certain molecules very effectively, like cardiac glycosides, and other molecules it doesn't adsorb at all, like monofluoroacetate in gifblaar doesn't work. Activated charcoal doesn't work at all for monofluoroacetate. We've dosed it before dosing the gifblaar, together with the gifblaar and afterwards. So we have got no efficacy against monofluoroacetate. Sorry, let me get this silent. Okay, so that's activated charcoal. They use activated charcoal when the gold price is high to recirculate these old mine dumps. It can even adsorb gold, which is a very small molecule. So it adsorbs certain molecules and some it don't. But remember, there's only one treatment for cardiac glycosides, 2 grams per kilogram body weight of activated charcoal, right there where you see the animal. You can repeat it the next day. We've done it this year with great success. Just leave the animal there. Don't inject it. Don't try and be fancy and inject it all sorts of things. Just activated charcoal. And in the tulp period in our practice, we keep about 50 packets of activated charcoal, and we sometimes use them in one day. If you have any questions on the cardiac glycosides and tulp specifically, you must post it and Helena will organize that for us. I can just tell you to adapt animals in my area, when they come from a non-tulp area, it's the plant with the biggest aversion factor that we are aware of. I put animals in a crush pen. I take some leaves of tulp. I put it in a liquidizer with a bit of water, make a slurry or liquidize the leaves. And I take a 5 ml syringe and I put it per os in the mouths of these animals for 5 days. And then they're used to the taste and they won't eat the tulp. I must say, when we put them on the tulp veld, we are ready with a few packets of activated charcoal. Instead, 1 or 2 animals might still try it. But that's the thing you can try with tulp, but not with other plants. The plants with the most active and prominent aversion factor is tulp. Okay, I've put something in for the guys in the Eastern Cape where we have lots of dairies. One of our 8 important plant species, Senecio. You can see, look at all the Senecios, and you can see that animal's liver is stuffed up. This is more Senecio than grass. The 2 toxic species in the Eastern Cape are Senecio latifolius and Senecio retrosus. It's a liver poison. And there's the Senecio. Usually they don't eat these adult plants. That's just for identification. They eat these growth stages and even smaller. But the adult plant, the flowering stage, are usually not eaten, which you can see there. Okay, severe liver involvement with, you get an acute phase of the disease where if they take in a lot, it knocks out the liver completely, and together with that, it knocks out the clotting factors. So what you see is not this what's on your screen at the moment. This is more the chronic form where you get nodular regeneration of the liver. It's only the liver that can do this. In the acute phase, you don't see this. It just knocks out the liver. You've got acute liver failure with bleeding throughout the whole carcass because the clotting factors are knocked out. This is a more chronic case where the liver tries to regenerate. So that's senesio. Quite interesting. Those of you who are in horses, this thing kills horses. Speak to farmers in the Eastern Cape, put a horse on senesio felt, it will eat the senesio, it will die. You can remember the disease is called Dinsiekte. If you read your textbook, they say the horses walk around aimlessly because the liver is so stuffed up that it can't detoxify ammonia, and you get hepatic encephalopathy. The ammonia affects the brain, and these horses walk around aimlessly. Let's go to another one, lantana. Unbelievably big problem in South Africa, invading the whole of KwaZulu-Natal, Eastern Cape, all our dairy areas, even the bushveld. This is a proclaimed week. It's not supposed to be on your property. And I had a program on television a week ago on this to explain to farmers how they must eradicate this. And there I've got a dairy animal for you. Okay, look at this. I'll take you back to your third year, secondary photosensitivity, which is very well shown here in a dairy animal because the unpigmented areas are more severely infected than the pigmented areas. And where can you see it better than on a Friesland animal? So let's go back to what happens. The liver is affected. Bile is not excreted, especially with lantana. It causes paralysis of the bile ducts. If you can remember how the secondary photosensitivity works, all green plants contains chlorophyll. Chlorophyll gets broken down in the rumen to phyloerythrin. Phyloerythrin is photodynamic. If phyloerythrin is not excreted via the liver, then and it accumulates in the body, it attracts the sun rays and animals become photosensitive. And that's exactly what you see there. It's a photosensitivity, sunburn in these animals. When I investigated this case, because animals are so etheric, the farmer treated these animals for about a week for anaplasmosis, gall sickness, because he saw these yellow carcasses. In the meantime, it was lantana poisoning. In unpigmented animals, the photosensitivity you only see on the muzzle here because this animal is pigmented. So be very aware of that. This is photosensitivity in a pigmented animal. You get severe rumen stasis, constipation, and really treatment. We use activated charcoal, but you get severe rumen stasis. It doesn't really work. These animals are gone. And unlike tulp, you will find that for some reason, animals will never learn to avoid lantana. We see it a lot there in Groblersdal, in the Loskop area, in the Loskop Valley. In the middle of summer, this time of the year, lush green grazing. For some reason, cattle one day will decide we're going to eat lantana, and they will eat lantana. So often we think that eating toxic plants are associated with droughts where there's no other greenery available. It's not so with lantana. I don't know why they do that. If you smell what lantana smells like, you can't imagine animals eating that. It's a cattle problem. And for some reason, they will just one day decide to eat the lantana where there's abundance of green grazing available. And then we get all these photosensitive animals and severe liver damage. You can't, and look at the gallbladder here. You'll see if you read your textbooks correctly, it say a lime, the gallbladder is overfilled with lime green bile. It blocks the small bile canaliculi in the liver, and therefore we don't get any bile flow, and therefore we get the severe icterus and bile stasis. Together with that, and we publish this, we also get severe nephrosis, which many people don't know. We consider lantana only as a liver poison. It knocks out the kidneys as well. So now you can see why we can't treat these animals. It's really the pathology is too severe. This one, plant Cestrum levigatum, you get Cestrum parkii as well, inkberry, was added to our toxicologist list of the 7 most important poisonings only in the last 2 years, because this plant is becoming a serious problem in South Africa. It's like 30 years ago when I started at OP, this plant only caused problems along the Vaal River. That's Botawil, Viljoenskroon, those kind of areas. Now it's widespread throughout South Africa. By the way, both of these plants, lantana and cestrum, are exotics. They come from South America. They proclaimed weeds. They are garden plants that escape from gardens. And look at this syrup here with the thousands and thousands of berries, and it gets spread by birds. The same with lantana, it gets spread by birds. So it's very, very difficult to contain. You need to go in aggressively and actively and continuously to eradicate these plants, because the birds will again spread them. The textbooks will tell you it's a shrub. If you speak to Dr. Olivier in the Eastern Cape, they say it's a tree, it's not a shrub. So depending on climatic conditions, it can really become a tree, and it sometimes plants against highways and that sort of thing, and all these berries and cestrum is spread into the farms, surrounding farms, and it's a huge problem. There's the liver, also again a severe liver poison. It was called Chase Valley disease, diagnosed near Maritzburg, where Chase Valley is. The liver is so knocked out that initially also you then get secondary nervous signs that the vets who then investigated the first outbreaks of cestrum thought it was rabies because of the severe nervous signs of this plant and the effect it has on the liver. So cestrum, look out for that one. Tremendously important. Now we get down to serious business as far as the dairy industry is concerned, and that's kikuyu poisoning. Maybe we will deal with it here. It can also be dealt under mycotoxicosis. We're not sure yet. The mysterious syndrome, that's all I can say about that. It's another mysterious syndrome which has no bearing or effect on dairies is geel dikkop. For some reason, sheep will eat dubbeltjies, tribulus for years and years, and just 1 year we get a severe outbreak of geel dikkop in the Karoo. And same here, animals will eat kikuyu as staple feed, the most common graze. And suddenly all hell breaks loose and you get these kikuyu poisonings. And there's 2 possible reasons for that, or 3. This is a typical pasture in the Eastern Cape, kikuyu pastures. And we get 2 syndromes. One is associated with armyworm and one not associated with armyworm. And there's maybe a common denominator between these 2 syndromes. So the syndrome associated with armyworm, we obviously get these worms. They migrate down from the northern tip of Africa with the high winds, Spodoptera exempta. This is the moth that comes down with the high winds from the East Africa, and they come with the high winds and they deposit the eggs on the grazing, and then we get these caterpillars breeding, coming out from these eggs, and they demolish grazing. And they come in their millions. And usually about 2 to 3 weeks after damage has been done to the kikuyu grazing, we get kikuyu poisoning. So what we think is that it's not the worms. We've dosed the worms. It doesn't cause any poisoning to animals. These worms must cause some sort of a plant stress, and then there's a secondary factor that comes into play when we look at these armyworms, and that will also tie into the second syndrome, which is not associated with armyworms. Okay, the worms themselves are not toxic. I said that kikuyu in itself appears to be non-toxic, but After grazing 2 to 3 weeks, we get these grazings, camps become toxic. And Dr. Johan Olivier, a well-known dairy vet in the Eastern Cape, he says the syndrome without the armyworm is more probable, and the armyworm syndrome is the exception rather than the rule. So they get many of these poisonings, and there were no armyworms involved. But the first documentation of the syndrome was always involved with where armyworms were involved. So there at the bottom of your screen in Australia, they also get kikuyu poisoning, and they isolated in these toxic pastures a fungus called Fusarium torulosum. And Professor Botha at the faculty also isolated this in South Africa. It's an endophyte, which means it's a fungus in between the cells of the plant. It doesn't occur on the superficial surface of the kikuyu. That's what we call an endophyte. It's in the plant. But and we'll come to mycotoxicosis a bit later, these things are very difficult to reproduce. Nobody is 100% sure that this fungus is really the problem. After these plant stresses, the fungus maybe takes over, but nobody is 100% convinced. But that is a theory at this stage. About 10 days, I said 14 days, about 10 days it takes to become toxic after these plant stress due to worms or not worms. And it stays toxic for 4 to 6 weeks after the worms has left. Okay, it's a gastrointestinal syndrome. So remember, we dealt now with TILP, which is a cardiac syndrome. We dealt with 2 liver syndromes, which is Senecio and Cestrum. Here we're working with a GIT, a gastrointestinal syndrome. So you get rumen atony, tympany, you get constipation, you get colic and constipation, but it's actually funny that you get constipation because you get a fluid-filled rumen. If you put your fist on that rumen and you press it in, you get a slosh slosh sound. There's lots of fluid in there, but the animals are constipated. This is a very interesting syndrome, sham drinking. It means they try to drink, but they can't drink. They can't swallow. You get that with rabies as well. By the way, it's a DD for rabies. Sham drinking because the brain is affected. But here's the major pathology. The rumens are filled with severely fluid-filled rumen contents. You see mostly kikuyu, slightly foamy, and that's all that you find in that rumen. And it's fluid-like, lots of fluid in the rumen. And that's where your pathology is. So you need to scrape out, clean out that whole rumen, and wash it off with a hose pipe with water. And if you take off the epithelium, here you see this severe rumenitis in the rumen and in the reticulum. So hyperemia, inflammation, and necrotic areas of the forestomachs. And you can take a sample of that and send it in for histopathology. Although these animals' rumens are severely distended and fluid-filled, they are severely dehydrated. So that fluid is not absorbed. And this is a disease with high mortality. You know, we get diseases like diplodia, which we'll come to later, which the morbidity is high. That's the animals, the number of animals that get sick can be fairly high, but the mortalities are low. This is a disease with high mortality. If they become sick of kikuyu poisoning, they die. Very few of them will recover. The treatment is symptomatic and supported, and there it says it, a high percentage of affected animals die. So there's actually no cure for this poisoning. We must, how do we prevent this disease? Prevent infestation of the pastures of these armyworms is fairly impossible with pesticides. They come in there, millions. You can test your grazing if it's toxic, and we'll come back to that later using your own animals. Put in a few tracer animals and see if the grazing is safe. Now again, 4 weeks after we don't see any worms anymore, that's the period. And then you can put in a few tracer animals. Then we get this ruminitis syndrome with kikuyu that's not associated with armyworm infestation. The pathology is the same. The whole syndrome is the same, but there were no armyworms. But there's maybe a possibility again of plant stress, and therefore this last paragraph is important. It usually occurs when there's a growth spur during warm weather, followed by high nitrogen fertilization and irrigation, and then stress on the plant thereafter. Again, the possibility of the fungus coming in. But you see, we're speculating here now. So we think about a fungus, and we'll talk a lot about fungi later. But there's some sort of a plant stress that make them more susceptible. It's the same with the, again, getting back to geel dikkop. Animals eat dubbeltjies, Tribulus terrestris, that's a staple feed in the Karoo for many, many years. Something happens, and then they become toxic. So these syndromes are very interesting and very, very difficult to research. Okay, we look at mycotoxins at the moment.